And that's it, everyone. Thank you for the great semester and have a great summer.
Okay, meet at the Rose Garden in 30 minutes?
Yes.
Sounds good. See you there! It's so cold! Why is it raining outside? It's supposed to be summer!
Why does the rain have to make it so freaking cold? It would be so much better if it was warmer. Why do we even need to feel cold anyways? Oh, that's right. I learned about it last year in Sensory Physiology Club. They said we feel cold through...
Hey, Melissa. Ooh, yikes. I didn't think it was gonna rain today.
I know, it's such a bummer. I mean, like, whatever happened to summer sunshine? But you know what, though? How about you say we go check out that new cafe across the street?
I'm down.
Cool! Last one there, the rotten egg!
Hey, wait up! Ah, it's freezing. It's so cold. I'm shivering. I would need a warm towel or something. How are you not cold?
What can I say? I'm built different.
Oh, that's cap. Everybody feels cold. I can even see the goosebumps on your arm.
Ugh, fine. You're right. But you know, there's this guy named Wim Hof. He could be in ice for like 100 minutes. And he doesn't feel cold. He just breathes and meditates and he's just fine.
Hold on. You're telling me we feel cold because it's all in our mind?
Not completely, no. I mean, of course there is our brain, but there's also our spinal cord, the skin, the nerves inside the skin. All of these things contribute to how we feel cold.
Well, aren't you smart?
But of course I am.
Now, come on, let's go.
This is my 1 year.
And now, let's meet our fabulous cast.
Katrina, come look at this cool video about Sensory Physiology Club.
Let's meet our fabulous cast.
Let's rock it. I'm gonna win.
Yeah, so they have this super crazy game show.
A game show?
Yeah, and you get to play as scientists, and these hosts, they ask you to do these experiments about how we feel cold. And you know what the best part about all of this is?
What?
If you get a question right, you get to dump a bucket of ice on the host's head, and then they get to suffer from extreme pain from the cold.
Ice? I'm convinced. And what's the prize once we finish all this?
Knowledge, Katrina. Knowledge.
That sounds pretty fun.
Yeah, and you know what? They just opened up applications for this year, so we
Really?
should apply. Yeah, we should apply and try to participate.
I'm down.
All right, well, let's do it.
Let's do it.
Oh my gosh, remember that crazy game show we applied to?
Yeah.
We got it! Yeah! Yeah!
We're gonna crush it.
Yeah, we will!
Action.
Hello, dear friend.
Action.
Hello, dear friends. No.
Cut.
Hello, dear friends, and welcome to our game show, Ice Buckets versus Scientists. Today we will help a confused group of scientists to discover how people feel cold. Together we will guide them through using the scientific method to obtain new knowledge about our universe. At the same time, we will explore our beautiful lab, our equipment, and we will learn how some of the common biological methods work. And now, let's meet our fabulous casts. Our journey today will take us through several stages of the scientific investigation, and if you successfully complete each stage, you will get to see how our scientists celebrate in a very special way. Believe me, it's worth. So pay attention, think, and participate. Every story has to begin, and scientific stories usually begins with a good question that people are curious about. Usually those questions are something like, how does it work? Or what's the mechanism? You have probably heard the expression standing on the shoulder of giants. This is especially relevant in science because through the history a lot of people have investigated their discovery about the world and reported. So when you have a scientific question, the first thing to do is to check what other people have already reported.
Scientists have found that cooling activates nerve endings in the skin, which belong to sensory neurons. These neurons then transmit the signal to the spinal cord, and ultimately to the brain cortex, where they create the conscious perception of cold. If we dig a little deeper, we will see that on the surface of sensory neurons, or in other words, in the plasma membrane of the cell, there are these proteins called receptors. They are like little sensors that detect different things like temperature and chemicals. When they detect cold temperature, they are activated. And when enough of the receptors are activated by cold temperature, the neuron fires and a signal is sent to the brain.
I hypothesize that the receptor M8 is responsible for cold sensation. If my hypothesis is true and the M8 receptor is responsible for cold sensation, then receptor M8 must be first, sufficient, and second, necessary for cold sensation. By sufficient, I mean that if we put the receptor in any cell and that cell becomes sensitive to cold, the receptor is sufficient for sensing cold. By necessary, I mean that the receptor has to be there for cold sensation to work. If we get rid of the receptor and cold can't be sensed, then it is responsible and necessary for feeling cold. Scientists call getting rid of a gene knocking out a gene.
Each neuron has genetic material made up of genes, or instructions, which make different proteins, including receptors. Genetic material is stored in a cell in the form of DNA. We can think of DNA as a recipe book, and the many genes or instructions which make each protein as a recipe. To transfer the cold receptor from a sensory neuron to another cell, we need to first learn the cold receptor recipe, or gene, from the sensory neuron. Since we don't know exactly which receptor is responsible for sensing cold, we will learn the recipes for the M8, A1, and C5 receptors. We can then give these recipes to a different cell, who is a bad cook, who doesn't know very many recipes. With the new recipes, the bad cook cell will learn to make the receptors for itself. At the end of the day, we will have 3 different cells that have learned to make one receptor each: M8, A1, or C5. Since cells can't talk, we have to use a different method to determine whether they feel cold. We will use an experimental technique called calcium imaging. When a neuron is activated, there is an electrical action potential. This causes calcium to rush into the cell. We give the bad cook cells that now express a new receptor a special dye that glows green when calcium enters the cell. We can tell when a cell is activated because it will glow green. Therefore, we can expose the cells to cold. If they glow green, it means they are sensitive to cold, and therefore that the receptor is sufficient to sense cold.
We will knock out our candidate cold receptors M8, A1, and C5 in mice and determine whether they can still sense cold. These are specially generated mice that are normal in every way except they lack one specific protein.